Fantastic. That's looking great. Well, welcome back to Neuroccino in this new term. Due to a bad internet connection, I'm just going to step back and let Valentina take over for the rest of today.
Thank you, Steph. Thank you, everyone, for joining us to the Neuroccino. Welcome back from the summer break. Here today we have Maria Petronovic, am I saying correctly? Who's going to present science. What's sex got to do with it? To you the floor, Maria.
Oh, thank you very much. So first things first, can you hear me? Can you see me, right? Okay, wonderful. So thank you very much for this invitation. And it was some kind of invitation was the type of, Maria, there is a great paper, absolutely fantastic story. You just have to present it. So I was given with the story, but the story then turned out to be absolutely a great one. And instead of reading one paper that I got, I was reading about 7 to 8 papers just because of my own interest, because the topic is really, really cool. And yeah, Steffi, you were the one forcing me into this. So now I know a little bit more about sex and science, and there are so many connections between those two. And when today you will hear me saying word sex so many times, and when I say sex, it is on purpose. I'm thinking about biological construct, and we are going to talk about biological construct, not a sociological one, which is a gender. So today we are really talking about sex, either of animals or experimenters, and what that got to do with science. So let's start. Can you see my screen actually?
Yeah.
Okay. Okay, recording progress. So some time ago, Steffi sent me this paper, this perspective on how we should actually consider sex as a biological variable in our preclinical experiments. And for Steffi, this was something very new, because most of the people that I work with actually work in clinical studies, while I'm a kind of a basic neuroscientist. I work with mice and rats. And for us, we knew for a very, very long time that female and male mice or rats are quite different. And very often we actually, based on the experiments, based on the question we had, based on the disorder we worked, we chose either kind of males or females. However, it turned out, and there is a lots of text because I was just taking it from the papers instead of showing a paper. However, it seems that vast majority of studies in the preclinical neuroscience field use actually males, and it seems that approximately males are used approximately 6 times more often than female mice and rats. Why is that bad? I'm sure that you all know the very infamous example of thalidomide. So this was a drug developed sometime in 1950s to treat morning sickness in pregnant women. For treating morning sickness, it was absolutely fantastic drug. However, it had a very serious side effects. It caused several thousand fetal deaths, and more than 10,000 children were born with different types of bodily malformations. You have surely seen pictures of that, babies born without legs or hands and so on. And all of that actually turned out that most of it could have been actually prevented by testing thalidomide on human female tissue. It was all tested in males, and nobody knew that it's going to be so teratogenic. So studying both sexes is something which is in a way mandatory, really necessary, but people don't do it for very different reasons. And those reasons are actually really highly detrimental for half of the world population, that half being women. Why is that? We now absolutely know that physiology, some parts of our physiology, not just the reproductive one, are very different between males and females, that we do not react on the same drugs the same way, that mechanism of disease are different, then the presentation of a disorder or a disease is very different. As they said in that paper, one of the most well-known examples is absolutely cardiac arrest, which in women shows different symptoms. So women actually die more often of cardiac arrest than males because, you know, when you arrive to the doctor, they have this book. They look into the symptoms there, and those symptoms were developed based on men's symptoms. So you don't fit inside of, but you don't have a stroke, and you are going to die most likely. So very, very similar is actually neuroscience. And you may actually know if you work in the field of autism or ADHD, but especially autism. Not just that autism is more prevalent in men. This kind of prevalence in males is constantly kind of shrinking, and females are coming closer. The more clinicians actually realize that females are simply underdiagnosed because definition of autism was done on male children. And female presentation is very, very different, but it doesn't mean that girls do not have autism, and they're often not treated on time, and that has lifelong consequences. So half of the world population is actually, you know, disadvantaged. So what did NIH, but this is National Institutes of Health in the US, follow the suit of EU and some other governing bodies, and actually said that this has to stop, and that we have to start using both male and female animals in our research. Easily said, but it's never easily done. And we will go to some of these reasons. So funding agencies, and I can actually confirm that many of the funding agencies, especially American ones, when you kind of submit your grant application, they want to see that you're going to use males and females. So, but why is that again a problem? You know, as many of the diagnostic criteria for humans are done on males, looking into males, also many behavioral paradigms that we use when we are testing animals are done on males, because a long time ago, people were using only male animals for experiments. So we know how they react, but is it now okay to use this test also for females? Or are we again going to get skewed data? It seems that at least in some instances, we are getting skewed data. So making the story short, for example, you all know that our hippocampus is incredibly important for spatial learning and orientation in space. And everybody who knows me well, no, even not that well, knows that I simply can't find my way in space. Reading a map, it's a science, you know, it just doesn't work in my case. But it seems that this is actually not so, it's not, you know, that I don't have a hippocampus or it doesn't work, but I use different strategy than males. And apparently preclinical studies have shown that. So it seems, as you can read here or in that paper, that, you know, based on many of those experiments done in the last few decades, scientists have very conveniently this kind of concluded that females are just not so good in spatial orientation than men because they need longer time to actually do the test. But it's not because they're stupid that they can't do it. They're impaired. It is just, it was found very recently when they were looking really more in depth into all of this, that females use a very, very different strategy to arrive. And one of those strategies is actually very important for females, much more than for males. And that one is to be sure that you avoid all possible kind of dangerous situations around you. Why is that more important for females, female animals, than for males? Pregnancy and having babies with you. Usually male animals, definitely male mice, don't have babies following them. So this, you know, this female mouse needs to be sure that she will be safe and, you know, her progeny will be safe. So very different strategies used. There are many examples like that. And I will just go into probably just one more. And this is something called conditioned fear learning. So what you do there is, you know, you have this box, testing box, and you usually give either a strong tone, very loud tone, noise, or you give even a foot shock. And how you know that animal is afraid is because the animal mouse freezes in spot, just freezes. And if any of you have ever been in a MRI room, when somebody started kind of imaging sequence, not knowing that you are inside, you just freeze because it's such a loud noise. It's so bad. So mice do the same, they freeze. But only male mice freeze. It seems that female rats, for example, do not freeze. They try to escape. Again, most likely having progeny or being pregnant with them. They just try to, And what was the conclusion for many years? Oh, they're not afraid. They just, stupidly go around. So we also need to use different tests. It's not just now including males and females. We need to actually think about different behavioral paradigms, but it seems that it's not only behavioral paradigms, that will maybe be even easy, but there is something different in the physiology. So we now know that there are, of course, small structural differences, volumetric differences in the brains of males and females. We're talking male mice and rats here. And we also know that structural plasticity, for example, in these animals, in those two sexes, is different. So stress. Women are more prone to suffer from stress-related disorders, okay? And we have, again, worked a lot on animals, and it seems that repeated stress, when we talk about all of those dendrites and so on in the brain, it seems that repeated stress elicits dendritic atrophy and spine elimination in the hippocampus and I think orbital cortex in males, while actually in their amygdala, those spines start kind of even growing more. Excellent. So loss of plasticity in the cortex and increased plasticity in the amygdala. Wonderful. We know lots about that mechanism, how it works, which drugs affect it. And then what happens with females? Exactly the opposite than in males. So using the same drug on a female patient, in this case, even a human female woman, it actually doesn't do anything for a female. If anything, it actually gets worse for her. So how can we reconcile these two things? Difficult. So what NIH, I can't give you really a prescription, but what NIH and EU Commission, Wellcome Trust, MRC are now asking from researchers in the last few years is that we need to include, as I said, both sexes inside. Many people are strongly opposing to that, and I'm sure you will be shocked when I say that 2/3 of committee members in those bodies are males, and they do oppose to that. It's so shocking. They actually oppose. Why would they do that? And so again, we are getting drugs which, in us do not work. Many researchers will say, let me just see. Oh, this is chronic pain. We can come to that one later if you're interested, but I will show you something about pain. Many people, me included, until recording stopped. I've heard that.
Yeah, I think Sophie is having problems with the connection, but go on, don't worry.
So there are many excuses why people don't do that. And I have used some of those excuses before. Female data will be more variable than data in males. Why is that? You never know if your female mouse or a rat is, for example, at the beginning of pregnancy. You sometimes, you can absolutely exclude it, but sometimes, things happen, like in humans. Then, of course, estrous cycle or their menstrual cycle, and the fluctuation in hormones. So people thought, this will affect their behavior. They will not behave constantly the same way. But, we also know that testosterone levels in mice and rats go up and down like in humans. So this is not a valid excuse. What could be, let me just see if I have it here. what could be potentially valid excuse, and is that including both males and females in your studies needs a lot of money and needs a lot of human resources. And somebody just starting their group, still being, the first stages of that academic career, simply cannot do that. I have seen big pharma companies who also didn't do testing in animals, for example, for autism or ADHD, because the manpower needed to do that needs to be immense. Analyzing behavioral data takes months. Not everything is automated. So there are lots of roadblocks. One of them is also kind of publication bias. So let me just see if it is on this one, on this slide. So if any of you works in a preclinical field, then when you submit your paper to a very good journal, magic word to pass there is that you have the mechanism of a disorder. If you don't have a mechanism, you don't go to Nature. You go to +1, okay? And in order to get the mechanism, you need to dig really deeper, so very, very deep. So you see how your animal behaves. Then you go into electrophysiology, then you go into optogenetics, then you go into MRI, then again into chemogenetics. Just this takes approximately 4 to 5 years for one project to be, started and published. Enormous amount of work. And then if you want that to be done in females, very, very difficult. And again, you need to publish this. You need to publish faster than somebody else. You need, your career, you need your title, how somebody's recently told me. And, you prefer such a paper than having a study done on males and females. So many things really do need to change in order that we will be able, or even willing, really, to do something which is proposed in this perspective. However, things are not that black and dark, and for example, what they omitted in this paper, and if you work again in basic neuroscience or immunology, whichever field, you will know that if you, for example, work in cell cultures, if the cell culture is embryonic or fetal, you don't know who is a male, who is a female. You always have a mixture. So a vast majority of cell work, if it is not on well-established like HeLa lines or something like that, which is a female line, it will be kind of mixed. But now what I would actually like to say is also, when I was just a couple of minutes ago saying, including both males and females takes enormous amount of resources, human resources. And why is that so important? I will tell you now in the next few slides. It seems that not only sex of the animals is important, but the sex of experimenters plays a role in the reproducibility of the data. And I will explain it later, but let's just now say that my new PhD student wants to, look into whatever aggression, like my students are doing. And of course, they look only in males. If I now want them also to look into females, one student needs to do all of that. All the behavioral data need to be done with one student. And that one student cannot do that within 3 years. End of story, cannot, just cannot. So do we do it to 2 students? If yes, can they be both? Can they be one girl, one boy? Or do they both need to be females or males? Oh, well, if you see this slide, this was published in 2014, Nature Methods, great stuff. And I remember that when we saw that paper in 2014, it was a little bit of a haha, but then we started checking our data. And the lab in which I was working then, we were mixed, men and women, and we actually didn't really see the difference in our data. However, this paper says that it's kind of that the data, that the results of a study are heavily influenced by the sex of the experimenter, meaning males or men stress mice and rats heavily. And I will briefly show that. So, of course, that potentially confounds the data. So what they did is that they injected kind of a painful stimuli, so inflammatory agent, into the ankle of the animals, and the experimenter was half a meter away from an animal. When I do experiments, or my students, we are never half a meter from the animal. We are kind of 2 meters away, just that they also don't see our shadow and so on. But they did that, and in the room, in that half a meter, were either male or a female, or the room was empty, or they even kind of just presented a male t-shirt, sweaty t-shirt from the gym, or a female t-shirt. And incredibly, those animals, male animals, who were in the presence of a man, actually were stressed. And how they knew that, first things first, something that I personally wouldn't really rely on, but it seems, it's nature methods. They checked the mouse's face, and kind of this grimaces. Mice actually do grimaces. We see that from time to time, but it's very subtle. You really need to be very skilled to see that. And they have seen that mice actually, you can see it, I think, here. So, of course, pink is a female, clearly, and these blue ones are guys, just gender stereotypy. So mice were grimacing a lot when the males were around, but not when the females were around. And then they actually went to check baseline pain levels. So how you do that in the animals? There are many different ways. You can pinch the tail, so-called tail withdrawal, or, for example, put it in a hot water, not really hot, or put their foot on top of a hot plate. Again, it's not hot to cause burns, or to use something from human field called Fonfrey filaments, so little needle kind of poking into their paws. And you can see that all the males actually have something kind of there was something analgesic in them, meaning that actually it took them longer to withdraw their tail or to withdraw their paws. And why is that? We know that actually in a whole animal field, there is stress, for example, causes this analgesic effect. So, for example, if you have ever watched documentaries of Sir David Attenborough, and you see a lion chasing the gazelle and then catching the gazelle and eating it, and she's still kind of half alive, but she never screams. And I remember always asking my dad when I was a little girl, why she doesn't scream? She's in pain. It's because of a stress and all the stress hormones coming up, that actually, you feel some kind of analgesia. There is no real pain anymore. And that happens with these animals. So if you look at here, these are the levels of corticosterone. And you see no exposure to an experimenter. So this would be kind of a more or less normal level. Look what happens to corticosterone when there is a male. What happens when there is a female, practically baseline level. If there is a sweaty male t-shirt, they go berserk again. 30 minutes, that same t-shirt 30 minutes later doesn't do anything anymore, because then mice get accustomed to it. And this is a female, this is a female t-shirt. And also something very ugly, but they do that. This is the number of fecal boli, so how much they put in the cage. Look, when they see this is normal, female t-shirt, normal, and they poo a lot when there is a man around, so they're really stressed. And very similar thing happens whenever there is a kind of a male of any species inside, rat, guinea pig, I forgot what else they were tested, or even a mouse, but a mouse that they don't know from before. If it was a cage mate, so somebody that they knew from before, like a brother or something, then of course there was no this effect. So what these guys in this Nature Methods paper concluded is that presence of male researchers causes stress in male animals, mice or rats, and this clearly affects their behavioral presentation, at least in pain research. They feel less pain. The same year, when somebody publishes something, then, if it is Nature Methods or any Nature paper, it becomes very fashionable. And suddenly people started looking into it in more detail. So they also use ketamine. You may know it as a party drug, like, most of the people know it. But this is also an aesthetic and something that these days people try to use as an antidepressant, because it really has very, very strong antidepressant properties, with, active within hours. It's really, really great. However, again, it seems that the sex of the experimenter matters. So apparently this ketamine, let me just see if I have I don't have the graph, sorry. It seems that ketamine showed antidepressant properties only when given to male rats by a male experimenter. So they put them into a swimming pool, mouse swimming pool, and they check how much they swim. And if you give them anxiolytic or antidepressants, they can swim much longer before they just lie down and, okay, I need to be rescued now, I can't do it anymore. But when female researchers gave them ketamine, the effect was not there anymore. Apparently, they did the same experiments over several labs, and many labs have actually showed this. And it was also publicized very highly a few years back. Also, T-shirt with sweat of the males and so on. Ketamine didn't work. However, there are researchers like Lisa Montejo, very famous researcher in this field, saying that, we have decades of research here and we have never seen something like that. And her argument is that it may be the presence of the male in the room, but definitely not who injected, man or a female. It needs to be a presence later on. And of course, they have seen, they put them, for example, they put the animals into the fume hood. After all of this had been argued. And in the fume hood, when they cannot smell a male or a female later on, nothing happens, which is, again, really, really strange. However, it really seems, and many people are now being aware of it, that sex of the experimenter can actually skew your results. And people are now asking for yet another thing from preclinical researchers. And that is that we need to now, when we publish our papers, that we should clearly state the sex/gender of the experimenter, which is, I believe, many people will probably agree with me, which is not just GDPR issue. But, as I said, when we talk about animals, we talk about sex, biological construct. When we talk about humans, we talk about gender, we talk about sociological construct, not the biological one. So reporting that could be very, very tricky and very not GDPR and very not politically correct. So at the moment, I really don't know how to go around this issue. But it seems that this is not an issue only in preclinical science, but in the clinical studies as well. And I was really interested when I started reading all of this, and it seems that the effects of researchers' gender or sex, when it was done, it was a long time ago, they were just using the word sex then, was quite well known in the fields of psychology for really, really long time. But still, when you read clinical studies, gender or of the scientists doing the study was never reported. So this is the paper that they have found, published in 2018, just kind of a very nice review, stating that actually part of the reason for low replicability of clinical studies, at least in the fields of psychology, could be, one of the reasons, again, just one of them, could be the gender of the experimenter. And they gave several very nice, very interesting examples. Of course, one again is pain. And it seems that, if you have males and females in your study, and experimenter is a female, especially when it is a very attractive female, males immediately have much higher threshold to pain. They're braver, whatever, and the threshold to pain is higher suddenly. This changes when you use the same participants, but with a male, when the male is there. IQ is another example. So apparently some tests were done on children, and if the researcher was a female, IQ scores were higher at the end of the testing than if the researcher was female. Why is that important? I mean, when we look into neurodevelopmental disorders, or where we look into learning disabilities and developing new drugs for the learning disabilities, you really need to know what kind of a learning disability that is. You rely on IQ testing. Alzheimer's disease, for example, is another example where this is really important. So you can really get false negative results. Let's say your drug, doesn't work just because your experimenter was of a wrong gender. So I think I have even put an example there. So imagine if males would test, for example, your participants, and females would test the placebo instead of a drug. You will get completely skewed data. However, things are never that black and white, but there is, I think, quite sufficient evidence these days that we really do need to pay attention to that. Oh, something happened with my presentation. Sorry. Oh, actually, I cut out the last slide. Sorry, I did it last night and I didn't realize.
That's all right. Time's up anyways.
Wonderful. Was I just on time?
Kind of. Why
did you just stop me? I didn't know what's the timing is. I was expecting, regular 45 minutes. So this is the topic for today. I know it's a little bit different than usual, maybe even funny for a Monday morning, but something that we definitely should keep in mind. And somebody asked, Steffi, some references were there, some are not. I can send the papers during the day if that helps. Let me just, Steffi, are you there? I will upload the papers right now.
Lovely, thank you. Whilst my internet is holding up, I got a question, if I may just jump in right now. But you mentioned that, so clearly the awareness has increased about needing balanced preclinical samples, and funders are requesting it. But how much has actually changed already? And what was the difficulties about having female rodents in your sample? I may have missed that due to cutting out earlier. Thank you.
I mean, as I said, I was recently applying for a grant in the States, and they definitely do ask that we should include both females and males. They really, really, really do encourage that. What I didn't say is that, but it is on the presentation, that those examples, this kind of gender influences have been seen in experimental gender influences, have been seen in rodents, but not in zebrafish. And people are now really pushing towards zebrafish. Of course, in water, they can't sense the smell, and so on. And I don't know how about animal gender there, animal sex there. People are trying, but as I said, imagine you have limited resources. You are assistant professor, associate professor. You're not, you don't have millions. It is, I would really argue, it is extremely difficult to do that, because it doubles the amount of work. And if you really think that the experimenter sex plays a role in the producibility of data, we need to either have one experimenter doing all the behavioral experiments in that cohort, or both of them need to be females. or both of them needs to be males. So how that affects our recruiting again? There are many issues around it. And of course, in some instances, it makes sense to do experiments only on male animals. If it is a heavily predominantly male disorder, this is how you will start, but then you have to move towards females. It is difficult. And many years ago, when I was doing my PhD, and this was spinal cord injury, I have asked my boss why we were not really using males at that time. And I said, okay, we really need to start with males. And the kind of answer was like, come on, male rats and mice, they're like human males. When they're sick, they don't do anything. I didn't say that. My boss said that, and he was a man. Okay. It's going to be difficult. And also, as I said, not just these resources, but if we need to go into a mechanism of a disease or of a disorder, you can't test in same study both males and females. It's just one study just on males, so just on females takes 4 to 5 years. I don't see, if you don't have 30 people in your lab, I wouldn't know. But
Thank you.
yeah, I think I have uploaded a bunch of papers. Leah?
Yes, thank you. It was really, really interesting. And I had like a kind of provocative question. So is it like an utopia to have a test or an experiment that will fit for both male and females? Or we are so different that there's no way, we have to be tested separately in different ways?
My gut feeling is that probably many tests would be fine. Like, for example, at least for mice and rats, like, for example, open fields. This is just a huge box. You put them inside and you see, how much they move or how much they like or dislike the light. You test locomotion and anxiety. That is probably going to be very, very similar. But then, for example, as we said, freezing, fear, it's different. I work on aggression and we never test females. I never did it until now because the aggression in females, we know that is very different than aggression in males. And it's mostly driven by the fact if the rodent, female rodent, has pups or not. If they have pups, they're very aggressive. This is normal. If they don't have pups, they're almost never aggressive, towards other females. And we still don't really, we have some tests, but not much. Territorial aggression testing that we do in males doesn't work in a female. They're not territorial, they cooperate, they can be together. So I think that many new tests will have to be developed. But how many? I was shocked by these experiments with spatial learning and hippocampus. And it's a qualitative difference at the end. They do it a different way. So do we have, maybe once when things get more automated than now, it's difficult.
Maybe I was thinking that maybe the results achieved, like the score, like a goal. So not how you go there, but if you can do that. So something that go around of all these differences. So maybe the experimental have to deal with that.
Yeah, we need to find a way, but just looking if they can do it. So if you just look how, if they can do it, like, for example, this freezing, females, apparently they don't really do it. Mm-hmm. They run away, it's kind of a, how they call it, freeze or flight. They fly, they escape. So, I will get a very false negative result if I measure freezing as a form of fear. Mm-hmm. So I think we'll have a lot of new things to think about.
Yeah, really tricky. Thank you.
Chris?
Hi, thank you for the presentation. It was really interesting. I have a question about the part where the sex of the experimenter was influencing the results and the behavior of the mice, for example. How many experimenters were used? Because usually Only 4. The
In
problem
nature
is,
methods, I believe 4.
but it's weird because usually because in that case, the effect which they were trying to measure is on the what the human is producing. So usually when you try to see an effect like that, you're using a lot of participants. So I wonder whether there was a result with these specific individuals and maybe not with the entire population. It's difficult to generalize an effect on something with only 4 participants, no?
I agree. I believe, don't take me now for serious, please check the paper. If somebody's checking the paper, I believe it was 4, but it's nature methods, we have to trust it. What many people argued at the time, and me actually included, was that it may be highly likely, it's not even just the sex of the participant, gender of the participant, whatever we now want to use, is the skill of the participants. What I would like to know is if those experimenters are at the same level of proficiency, seniority, experience, how many years of experience working with animals they had. Because, I have seen some of my students, taking a mouse for a tail like that, and my hair goes up when I see that. Mouse is stressed beyond measure. But the experimenter is afraid of that mouse. Like, I'm afraid of rats. I avoid working with rats. I think that my stress will stress them, So it would be really, really important to find out, I don't put, and I'm sure most of the PIs, you don't put a student to work with an important experiment. Just, I show you once how to do it and you just do it. We all have to have special licenses and so on, but again, some people are more skilled than the others. And I have seen papers which are not reproducible, mainly because of the skill of the experimenter. I think that this kind of a gender issue is very thought-provocative. There might be something in it, but I think we are far away from really, and really knowing. And if we really want to now include even that into our statistics, oh my God, number of experimenters, number of animals, but it seems that more and more people really kind of believe in that.
Also, I guess it would help to know what mechanism would influence the results, because then you could actually know, like if it's something in the smell, but you said there was an experiment where they tried to isolate the mice and they didn't find
there was no
effect.
Yeah, there was no effect.
That's weird. What else could so maybe the skill of the experimenter. So in that case, maybe that's why they found a difference, because they only used a very small sample of experimenters.
I mean, it could be, we are talking about evolution here now at place, and 2 males of any species, usually any species, they don't like being together. This is a fight for females, for territory, for food. They don't like each other. And, how they usually recognize that this is another male, not by looking at them like me, but by smell. And it could be that, of course, they simply smell a human male inside. It really raises the corticosterone, kind of, oh, now, there is a danger around. And that causes many, many differences. But the question is, now, does it really affect, as they said, only pain responses? Does it affect maybe locomotion? Does it affect anything else? We don't know. Really don't know, But I can tell you, male mice don't like each other, but experimenters have no idea. Only thing that I can vouch for, and I will stop here, is that years ago I was working with rats, and I was injecting them with something. And I was doing that without any problems, although I'm scared of them. They never attacked me. And then a technician, actually 2 technicians, came to me, both guys actually, and kind of were very angry at me that why I didn't tell them that my rats are aggressive towards experimenters. But they are not. they're caged with other males. They were not biting me, but they were biting them. But I don't know, is it because they are guys or because they were just, grabbing him and eject? I don't know. Valentina?
Thank you, Maria. My question is kind of related because I was wondering, you cited during your presentation both hormones related differences and like hormonal level and brain structure related differences that may cause different behavior and different biological reaction in mice. And also, I'm guessing in the Nature Methods paper where they presented with the sweat t-shirt. And so I was wondering to what extent if it is hormonal differences and brain structure differences. And is there, if you know, any research experiments with maybe transgender population who are undergoing like hormonal treatment?
Yeah, I was just waiting for this question. I actually don't have any answer to that. It's not really my field of research. So I was also new to topic and I just started reading about all of those stuff. But actually, I don't know. But as you have just said, and I have alluded previously, it raises so many questions, especially in human society. With animals, it's kind of easy compared to us. So let's say that we really need to report the gender of the experimenter. How do we do that? What is the situation, as you said, with transgender people? I don't know. I honestly don't know. I think it would be, I would probably oppose to reporting these things because this is GDPR. We see the authors list, but anything more than that, it's a little bit, it's something should be probably left to the discretion of a researcher. We just probably would be enough to say gender of the experimenter was taken into account, something like that. But I don't know. I honestly don't know. But I think this is an area where we really need to do a lot of research. Because if all of these things are true, and especially when I started reading about clinical studies, where they said that the gender of the experimenter there in psychology research, the effect of gender there was known for decades. How do you deal if you now have a person who is transgender or non-binary, or if a participant is homosexual? How, do we also now need to control that when we recruit participants? I don't know. I honestly don't know. But I think it's really important to start thinking about it.
Thank you. I think our time's up, and I think Steffi left us because of her bad connection. Such a shame. But yeah, she's clapping. Yay!
Okay.
Is
thank
there
you so much.
Yeah, I was asking if there's any other questions. You can either drop a comment on our YouTube channel, or here you can just drop a comment here in the chat, and then we can keep going chatting after we're off live. Yeah, thank you, Maria. It's been a great pleasure to have a scientist from the Kings. And I encourage anyone to join us in our Monday morning Neuroccino. It's not mandatory to have a presentation. Maria did a great job.
I know, it's just
but
easier.
You can just yeah, just do it as you feel. You can just bring a cup of coffee and the paper to have a chat on with us. So again, thank you guys. See you next Monday. Bye.
Bye. Thanks for the invite.
Bye.
Thank you. Bye-bye.
Bye.